Thanks, Ralph. It's really nice how these three talks have gone together. It's sort of like the first one focused on intelligence in terms of the logic pathways, and then we moved on to Tad's talk, which was focused more on information, and now with Ralph, implementation. So it's kind of I cubed here, intelligence, information, and implementation. Very, very nice flow. And that leads us to, I think, what will be somewhat of a synergizing talk on the subject of the pathways here from Renata Bushko. Renata holds a Master of Science degree from MIT, as well as a bachelor's from Smith College. She completed graduate work at MIT under the instruction of Professor Marvin Minsky, author of several books on AI, and a founder of the field of AI. Ms. Bushko founded, after that graduate work, the Future of Health Technology Institute, and currently serves as its director. She's chaired now a dozen Future of Health Technology summits, which focus on engaging leading minds from the technology and health fields into basically appreciating and further developing the future of technology for the benefit of global healthcare. Renata, please join me at the podium. Thank you.
Thank you very much for the gracious invitation to be here today. Thank you to Dr. Rothblatt, and I also wanted to thank for a great dinner conversation yesterday to Dr. Bloomberg, who inspired me to mention a couple of things about space, so you have to bear with me. Today's topic, Chromollacytes and diamond mechanosynthesis, will be covered in the following way. I'd like to start with an introduction and mention a couple of words about Future of Health Technology Institute. Then go to describe and introduce chromollocyte, introduce basic concept, design process, selected components, and pretend that we are replacing our liver in vitro with all those, with many chromollocytes. Eventually in Act 2, and I think it's truncated on the board here, in Act 2 we'll look briefly at diamond mechanosynthesis, and in Act 3 we'll talk about significance, and I'll give you some references to learn more, and I think learning more about chromollocyte design and thinking about it further will be something that is very beneficial. Now on the right here you see a picture of the space elevator, and I use this picture because a couple of years ago I invited space elevator scientists to talk at one of the Future of Health Technology summits, which for the last 12 years for me are the basic brain research laboratory mentioned by Dr. Rothblatt. So it's been a great pleasure to meet many interesting scientists over the last 12 years, and one of them was Dr. Edwards. Space elevator brings an idea of science fiction coming to life. It was imagined and dreamed of by Russian scientist Ignacy Tsiolkovsky. He thought of celestial castles, that's how he called it, celestial castles. And now, of course, it was mentioned by Clark too in his book, Fountains of Paradise. Thanks to carbon nanotubes, the idea becomes more and more real. So the space metaphor is important to all of us who are trying to bring concepts from dreamland to application world and to reality. Now, it's also important because we're dealing with when we think about space, we deal with universe, huge universe. Now, we are all talking now about nanotechnology today, so it's also a universe, but universe of our bodies. It's the same kind of thinking that can be applied. Mission of Future of Health Technology Institute is to bring together creative minds for Future of Health Technology Summits and do exactly what Dr. Rothblatt mentioned about today's meeting. The two books that you see are the result of those meetings, Future of Health Technology, published in 2002, and Future of Intelligent and Extelligent Health Environment, published in 2005. I'm working on the third volume called Strategy for the Future, and I'll tell you more about it during our social time, I guess. Well, the goals, I believe goals of Future of Health Technology Institute are shared in your hearts, so let's investigate them. Stop disease before it even begins. Stop suffering before tears occur. Stop symptoms before they hurt. Stop medical errors and aging before they kill. Stop cyborgs before they control us, on the inside or the outside. Future of Health Technology Institute tries to achieve this by beginning to address health crisis as a national, international emergency, and we are in crisis. Beginning to think about long-term global future on an ongoing basis. Beginning to manage randomness in technology creation and adoption process, not removing randomness, but maybe slightly decreasing it. Beginning to design intelligent and extelligent health environment with caring machines. Annual Future of Health Technology Award is in the form of Pegasus. The wings symbolize inspiration, and by my training with Professor Marvin Minsky, I have to say that logic is not everything that we should be thinking about. Leap of faith, guessing, that's why wings are there. Now, there is also a third eye that Pegasus has in the middle of its forehead. And the eye is a diamond eye, nanotech-made diamond eye. It symbolizes the ability and the willingness to see the future and to open to the possibilities that the future brings. The recipient of that award last year was Ray Kurzweil, and he gave a wonderful talk how human potential can be increased with technology. The vision of the future here is very simple. We have equation, life science plus computer science, and all the convergence that you see of mathematics, molecular biology, mechanical engineering, electrical engineering, biology, nanotechnology with prominent feature of Chromollacyte listed here, and genetic engineering. It's all merging, and the result will be hopefully decreasing the speed that we age in and a new life form. Every year we meet and we think about a very important question, and I think on personal level, each and one of us thinks about it, how to give ideas with the biggest impact a chance. We have limited resources. It's good to imagine that we have unlimited resources to actually get that question answered. But how to give the ideas with biggest impact a chance is a very important question. Now I'd like to introduce Dr. Freitas, Senior Research Fellow at the Institute of Molecular Manufacturing. I had a pleasure of meeting him over the internet. He contributed a chapter about nanomedicine to my first book, and I've known him now for 7 years. I believe his work is incredible, and I call his paper on the ideal gene delivery vector, Chromollacytes, cell repair nanorobots for chromosome replacement therapy, a historic event. It's worth reading the whole 50 pages. The first theoretical scaling analysis and design of a cell repair nanorobot is in that paper. I also believe that the work presented by Dr. Merkel jointly with Dr. Freitas is of historic significance. And I just want to mention, by comparison, a paper written by Schrödinger titled "What is Life?" where he predicted DNA, basically. And I'll give you a couple of quotes on the philosophical end here that emphasize the chromosome importance. The most essential part of living cell, the chromosome, may suitably be called a periodic crystal. In physics, we have dealt only with periodic crystals. Compared with aperiodic crystal, they are plain and dull. aperiodic crystal, in my opinion, is the material carrier of life, and DNA was not discovered at this time. The question here that Dr. Rothblatt mentioned, time and space, which I thought was very, very important. We keep thinking in a greater level of detail, but here we are, time and space. How can the events in space and time, which take place within spatial boundary of living organisms, be accounted for by physics and chemistry? That's what we're all about. That's what we are thinking about. And on the optimistic note, the quote from Schrödinger, the obvious inability of present-day physics and chemistry to account for such events is no reason at all for doubting that they can be accounted for by these sciences. So any skeptics should read that twice. Well, Act 1, and it's time to meet Chromollacyte. You'll see a picture on the next slide. The basic concept is that Chromollacyte is capable of cellular chromosome replacement. So we have factories of good chromosomes, we load them on Chromollacytes deliver them into our bodies, and all genetically based disease is gone from the surface of the earth, and aging process can be stopped. On the slide here you see a nucleus and where the chromatin, the substance replaced by Chromollacyte, is located. And to put you in the mood, the chromosome is on the right. Chromosome 11, very disease prone, just to, as Dr. Merkel mentioned, you don't have to read everything on the list, but it just shows the complexity of the chromosome. Now, Chromollacyte design. Let's pretend that we're Dr. Robert Freitas now, and we'd like to design the actual artifact. Well, he said it would be good to minimize volume of the tool. Chromollacytes are not supposed to be free-floating robots. They will not free-float in the bloodstream. So the limitation of 4 microns, which is the limitation size for the free-floating robots, does not have to be maintained, so they can be bigger than that. But they are restricted to vascular surface travel, so they should be smaller than white and red blood cells. And they should be less than 1% of typical tissue cell, and they could be up to 25% of nucleus volume to be able to penetrate cells. Here we are. That's a hypothetical representation of Chromollacyte with dimensions of 3 by 4 by 5 microns. And that's from Robert Freitas' paper. And the close-up. Now you really met Chromollacyte. Hypothetical nanorobot. Chromollacyte has 10 basic components: probiosis manipulator, mobility system, funnel assembly, chromatin storage, power supply, navigation and communication system. There are computers on board, sensors, consumables, and of course structural support. I was interested in the mobility of the robot, so I focused on just a couple of features. Again, it's a 50-page paper that has all the details in it. So it's important to imagine to analyze the terrain in which Chromollacyte will be walking. So I call it the glycoprotein forest. So it has to be designed to get through that glycoprotein forest. The thickest glycoprotein forest is in our intestinal cells. Now, looking at the differences here, it would be nice to think how to proceed with Chromollacyte development. I would imagine that experts on various parts of the red cells, bladder, lymphocytes, intestinal cells, if we had groups of medical experts working with Dr. Freitas, imagine how much progress we could do, because there are intricacies of each and every environment in which Chromollacyte could function. Well, to make Chromollacyte mobile, Dr. Freitas installed 1,027 telescopic grapple manipulators on it, and they can hide and be extended. They can have different endings. They can bend. So you can imagine it's nearly like artificial cell that can move very gracefully. Because of that design, it can penetrate vascular walls, it can penetrate cell membrane, and most importantly, it's capable of penetration of nuclear membrane. Here I'd like to read a quote from his paper. It can also go through acellular tissue spaces and has to do that. A branchiating nanorobot can pull itself along individual fibrils, changing direction at fibril junctions, indirectly working its way towards its cellular target, crudely analogous to path of a sailboat tacking into the wind. That's so you can have a minute of relaxation here. Well, of course, all the design issues mentioned by previous speakers come to bear here. What if chromallocyte gets lost? What if it wanders around where it shouldn't? Well, there is a mechanism for that, but to find the next available fibril to attach to. But in general, coming from the area of artificial intelligence and working with Professor Minsky, I keep thinking about how to give chromallocytes common sense and how to make them adaptive. And that's a very important issue for all the nanorobotic design, and how to actually evaluate the level of adaptability that we are able to give them. I spent maybe 3 years on thinking about the measurement of adaptability, how we can do that. So I believe before we actually apply them to human body, we should solve that problem. Couple of elements, other elements of the design of chromallocyte proboscis is the major working unit that collects all chromatin from the cell and places the new chromosome, defect-free chromosome, in the cell. There are 10 CPU and memory units, 50 megabits of memory on board, and 10 acoustic message receivers. Eventually, the power supply is important. It comes from 10 acoustic power receivers, and patient is well coupled to a medically safe ultrasound traverse plane wave transmitter, so acoustic power is used. There is also buffer power supply in the form of 10 diamondoid flywheels. Now it's time for adventure. We'll pretend that we're replacing entire the cells, the chromosome in cells of the entire liver. In order to do that, if the basic procedure takes about 7 hours. We have to do organ survey and create a grid. It's one-to-one mapping of nanorobot targeting specific cell. That's the telemedical part, I guess. We have to prepare chromallocyte, load them with the chromosomes that we'd like to put in. Patient preparation involves actually applying respirocytes to make sure the pulse is down and that oxygen supply is good. Chromosome replacement takes place after robots are applied, and then post-operative process begins. The liver consists out of 250 billion cells, and it might require the infusion of about 1 terabyte of devices. So it's a huge operation, but it's possible with 63 cubic centimeters of chromalocytes dissolved in 1 liter of 7% saline suspension. And liver cells often have multiple nuclei, so we may have to repeat the entire procedure. Well, patient preparation is here important, and I work a lot on the macro scale in healthcare. So I focused here on what the patient, the human being, will be going through while chromallocytes are applied. So patient is placed on the ultrasonic vibrating table. Then patient is sedated and respirocytes are injected. Pulse rate has to be decreased, and then self-guiding flexible tube is installed and directly into blood vessel nearest to the liver. Robots then are inserted to the body, and it takes about 3 hours for them to do the job. To close, chromollocytes can only be built if we have the tool set for diamond mechanosynthesis, and Dr. Merkel addressed that in his talk, so you'll see the same tool sets again. To talk about significance of chromollocytes, I made a quick call to Aubrey de Grey, who actually will be visiting us in September, and that's what he said about chromollocyte. I don't think we're all that close to the operational chromollocyte, no, but we should be able to create such things eventually, and they will certainly give us radically new alternative approaches to combating aging and other medical problems. And the chances are high that some of those alternatives will outperform the more traditional ones. And the resources for more information about chromollocyte are listed on this slide. Dr. Frittes's paper is available for anybody who would like to read it. And eventually on September 22nd, we'll have another Future of Health Technology Summit. So write down the date, and maybe we can meet again and talk again about the future of health technology. We'll have United States and European Community Future of Health Technology strategy being discussed by experts, so I think it will be interesting. Thank you again for your time, and enjoy reading about chromollocytes.